Communities are open. All the parks are open. So it's kind of the kickoff to the summer season.
Okay. Well, it's already 7 o'clock, Pak Nazir. So here we have
with all of participants.
So we have host here, Nazir. And so everyone is coming. Good.
Banyak orang.
Yes. I invite my student to join this.
Good.
I'm teaching plant protection this semester, so maybe I have 140 students will join us tonight, I think.
Oh my God, 140 students. That's a lot. And I see Dr. Hasib here.
Yes, thank
Muhammad
you for coming,
Hasib.
Dr. Hasib.
Hi, good evening or good morning to you.
Thank you, good evening. Thank you, good to see you.
Nice to meet you.
Yes.
Good morning, Rob, how are you?
Fine. How are you?
Good to see you, excellent.
Hi, Sisi. Thank you for coming too. So, this Muhammad Hasib and Dr. Legaspi, my supervisor during my stay in the US.
Okay. Nice to meet you, Dr. Legaspi. And
nice to meet you too.
Thank you. Nice to meet you.
Dr. Wati said he will also join. She knows you. Bahagyawati.
Oh, of course.
Yes.
An old friend. Oh, she's going to. Okay, great.
Yeah, she said she will try to join.
Okay.
And I passed on the webinar to other colleagues too.
Okay. Okay, are we going to start or I hear there's some music here. Maybe we can have it stop. Yeah. Okay. So let us start. I would like to start with saying good morning and good morning for our colleague from United States, and good evening to all the students and colleagues from Indonesia, and maybe there are also some from other Southeast Asian countries. Welcome. It's a pleasure to host this webinar today. So the webinar, we titled the webinar as a journey of the Spodoptera frugiperda from Americas to Indonesia. Okay, and today we will be having 3 prominent speakers. The first speaker is Robert Meagher. I'm sorry if I say the name wrong. Meagher.
It's an unusual name. It's Meagher. We pronounce it Meagher.
Meagher. Okay, Robert Meagher.
But if I was in Ireland, I would pronounce it Mar, like Marr. So.
Okay.
Just call me Rob.
Rob. Okay, so the first speaker will be Rob, and then the second speaker will be Pak Yayi Munara from IPB University, and then the last speaker will be Ihsan from Universitas Muhammadiyah. A little bit introduction about Rob Marr. Okay, so let's see. He received his PhD from Penn State, and then master from Kansas State University, and BS from Shippensburg State College in Pennsylvania. And Rob is from USDA and have been active in ARS, Agriculture Research Service, since 1996, if I'm not mistaken. Yeah. Mm-hmm. So it's a pleasure to have Rob here, and for the next 20 to 30 minutes, we will give the time to Rob to learn from his experience on genetic variation of Spodoptera frugiperda. So, Rob, the time is yours.
Recording in progress.
Thank you for the introduction, and let me get my screen correct here. Can you all see my slides?
Yes.
Okay, good. So yes, I'm going to talk about genetic variation of fall armyworm mostly related to host strains, and then talk towards the end of my talk about some of the natural enemies. I would like to acknowledge my co-author, Rod Nagoshi, who actually has done all of the molecular biology and genetics work that I'll talk about here. So this morning or this afternoon, depending on where you're located, I'll talk a little bit about the history of the host strains, some of the molecular techniques that are used to diagnose the strains within North America. And then, like I say, I'll talk about natural enemies of fall armyworm at the end. The mid-1980s, and we're at LSU in Louisiana, and that's kind of at the head of the Gulf of Mexico. They knew fall armyworm migrated into their state, but they didn't know where the populations were coming from. So they had samples from all along Mexico, Louisiana, Alabama, Florida, Georgia, and then they got a sample from Puerto Rico. And what they were trying to do was to see the populations in Louisiana, how similar or dissimilar were they from some of these other areas. And what they found, instead of a geographical difference, they found a host strain difference. In other words, what those populations were feeding on. So everybody in the US, the larvae from those populations were all feeding on maize or corn, whereas the population in Puerto Rico was feeding on rice. And so that's where they saw the difference. They then went into Louisiana, started sampling in grasslands, Bermuda grass and other pasture grasses, and found the same similarity as to the rice. So that's why there's a rice strain the larvae predominantly feed on turf and pasture and rice, and a corn strain that feed on corn or maize, sorghum, and cotton. But those aren't for sure. I mean, you can get rice strain from corn and corn strain from rice. So now they're just called C strain and R strain. These strains were morphologically identical, so you had to use a molecular technique, a marker, to separate the two. And these two kind of go into the theory of host races, where these species where this one species is dividing, becoming sibling species eventually. Now, the markers that they used, and I'm not going to talk a lot about these. These were some of the older markers that were used to separate strains. Pashley's group used electrophoretic variants between the two. AFLPs were also used, and tandem repeats or genomic markers were used at one time. And actually, recently, Ashley Tesnow at Texas A&M University has used PCR of SNPs. But the two main markers, one is a mitochondrial marker on the COY1 gene, separating corn strain and rice strain. And the interesting thing here is with corn strain is that you actually, at two of these base pair locations, you can get 4 different haplotypes. And so the C strain can be subdivided into 4 haplotypes depending on the nucleic acids that are at that site. And so each moth has one of these haplotypes, but as a group, if you have a population of, say, show of how many are H, and the common ones are H2 and H4. And what Dr. Nagoshi found was that if you looked at samples in Florida and then you looked at samples from Texas, they were different, and the ratios were almost opposite. So the Florida had a ratio that was totally different than the ratio from Texas. And so the way we use these was we knew that fall armyworm overwinters. It cannot diapause, so at least in North America, it can't. We kind of put the overwintering zone around here. I mean, in warmer winters, it might be here. Colder winters, it's down here. And in fact, one of the worst years for fall armyworm was 1977, when it literally froze in Miami Beach, which is down here. And that's extremely unusual. And so the populations in Florida only survived down here. And for whatever reason, that next spring was perfect, that the populations exploded, and it was the highest damage ever recorded in the Southeast. But anyways, in normal years, this is roughly the latitude of overwintering. So we know they came. We know that they migrate out of these areas, but we didn't know where they went. Where, do they go over here? Do they go over here? Where do the Florida ones go? And it's funny. It gets back to the same question that Pashley's group at LSU was answering. Where do populations come from when they enter Louisiana? Whoops, sorry. There we go. So what we found, what Rod found, was that you could separate the Texas from the Florida moths. So we got a grant. We got lots and lots of cooperators from all over the central, eastern, and even into southern Canada to send us populations so that we could do the DNA analysis. And so what you see here, all the red dots, their natal origin for that one season came out of South Texas, northern Mexico, whereas the blue dots came from Florida. And the purple dots here are actually, that population is kind of a mix of Texas and Florida specimens. So you can see, and this is a kind of a diagram, that this is the Appalachian Mountains from northern Alabama, actually go up into Maine. But for what I want to explain here, we think they kind of form a barrier. So the Florida populations pretty much stay along the coast, and they actually kind of die out up here. They really don't get much further north. The Texas populations infest the whole central U.S. and then into the northeastern U.S. They even lap around sometimes and head back south. And then these are the ones that get into Quebec. They actually get in along the St. Lawrence River in Quebec, late in the season. So all of this work is, over 4,200 moths that Rod did the DNA for. We use for strains is the TPI gene. And I know this is a very complicated slide, and I will make this presentation available as a PDF to anyone that wants it, that they can take a closer look. And actually, Rod and I have a paper from 2022 that really explains this well. But this gene has a lot of variation. There's an intron here, the fourth intron. There's a lot of variability in this intron. So he can tell, he can separate host strains based on this technique. The difference is, compared to COI1, which is a genomic. This is on the sex chromosome. So you can get kind of two different views of what that population is. And actually what he did, what we did in this, and this is kind of a complicated graph, but what it's showing is what is the percentage of C strain that's in the population. And depending on if you look at it by the marker COI1 or TPI. So what he did, these are samples from all over the Western Hemisphere. USA, Brazil, Argentina. All these samples are from the Western Hemisphere, and in almost all cases, and this is in a corn strain, this is almost entirely from cornfields. What you see is that the TPI is actually a better marker at saying what is a C strain versus COI1, the mitochondrial marker. The reverse is true for the rice strain. There's no difference here. Now, obviously, the R strain, there's not going to be as many R strain in a corn habitat, but this is in R strain habitats, mostly pasture grasses. And so there's no difference between the markers. And then in kind of in the middle, a haplotype that in peanut can be both C strain and R strain. Again, you get a predominance, an improvement in strain identification using TPI gene. So even though there's a strong correlation between the two markers, the TPI marker detected a higher percentage of C strain than the other marker. And there were statistical differences in C strain hosts. So this is the final slide I want to talk about with genetic variability. And it's kind of an interesting slide in that, it shows that fall armyworm is not a newcomer to the Eastern Hemisphere. It's been found several times before. It was found in the 1960s in Israel. They actually have genitalia mounts of it, so they know for sure it was Spodoptera frugiperda. They put traps out, but they never found it again. So whatever it was feeding on, it died out and was never found again. Germany in 1999, it was a kind of an odd case. It was one, wasn't shown up again. Vietnam, there's reports that it was found on turf grass in Hanoi in 2008. And the most interesting, and this is a little controversial in the sense that we don't have any of these samples, but USDA APHIS has people at all the major ports where agricultural crops, cut flowers, any kind of ornamental plants come into the ports. And they take a subsample of all those plants and look for what invasive species might be coming in. There was a report that prior to 2014, Spodoptera frugiperda was found on produce or on plants that came from all these different countries: Turkey, China, Indonesia, Israel, Micronesia, Netherlands, and Thailand. And again, we don't have those samples, so we can't go back and do DNA. This was all based on someone looking at the larvae and identifying the larvae as Spodoptera frugiperda. If this is true, then that means that Spodoptera frugiperda has been in the Eastern Hemisphere for a long time at different levels of infestation. Obviously, 2016 was when it was noted here in West Africa. And if we look at the Texas, Florida haplotypes that Rod came up with, you can see clearly the initial populations were the Florida haplotype, and then most of Africa is the Florida haplotype. The Texas haplotype did come in 2018, but in small numbers. And even here in Asia, where Rod has gotten some samples from different parts of South and Southeast Asia, again, they all come up as the Florida haplotype, compared to, say, South America, where everything is the Texas haplotype. And so there's recent research out of Australia that hints that perhaps instead of a west to east migration of fall armyworm, actually there's been several introductions in Asia, and populations have actually moved to the west. So this, there's a lot more research that needs to be done to clarify that. But again, it's kind of interesting. Spodoptera frugiperda, even though it's from the Western Hemisphere, has been in the Eastern Hemisphere at different times. Okay, so I'd like to switch gears to natural enemies. This is some work, and in Florida, fall armyworm is a real serious pest. In South Florida, there's one of the counties in southern Florida that is the third largest producer of sweet corn in the nation, and it's fresh market sweet corn. So that means that the ears and the kernels have to almost be perfect to be sold. It's sold at a time of the year where there's no one else in the country that's making it, so it's very valuable. It's a high price commodity. So fall armyworm can be a serious pest. This is some work I have done through the 2010s, 2012 here, 2016. This work, and we have two main parasitoids, Cotesia margin of interest, which is a larval parasitoid, Chelonus insularis, which is an egg larval parasitoid. The female stings the egg. The egg of the wasp stays within the molting progeny, and about third to fourth instar, it finally hatches out and devours the fall armyworm. So what I found here, and the corn, the only way they control fall armyworm in corn or sweet corn is through insecticides, and different growers will spray at different times of the corn crop, and so some will even spray fairly early. And what I found was, interestingly, so I sampled fields that were sprayed and unsprayed, and with Cotesia, there really wasn't much of a difference. They seem to not be bothered by the insecticide sprays. The parasitism level was about the same. However, Chelonus really was affected by the insecticide sprays. And you can see here in the sprayed fields, hardly any Chelonus at all, whereas in an unsprayed field, very high levels of parasitism. And you can look here, this is work just from a one season, spring season in 2012, all in southern Florida locations. And Chelonus at times can be a really have really high levels of parasitism. I had one field, and I'm not showing it on this graph, that we had over 90% parasitism from Chelonus. So if you don't spray early, then you can actually get a lot of control from this parasitoid. Of course, the other parasitoid that a lot of people have worked with all through, they're working on it now in Africa, but all through Asia, is Telenomus remus, egg parasitoid, really little guy that stings the eggs. The story in Florida is actually really interesting. So I call it Florida Releases 1.0. So the first releases were in mid-1970s. And so the populations came from Malaysia. They were established in Barbados, and then those populations were sent to Trinidad. Unfortunately, the populations were not, they were kept in a laboratory, but when released in the field, they never established. But they moved some of those populations to Florida, South Florida actually, Homestead, which is south of Miami, and released them there. They also put a colony up in the Department of Plant Industry up in Gainesville, a state of Florida quarantine facility. But they went, after the field releases, they went back out. They never found Telenomus, so it never established that year. Florida 2.0, they tried again in the late 1980s, releasing 110,000 specimens. These came from established field populations in the Cayman Islands and in Puerto Rico. And again, were released in Homestead in South Florida, and again, never found them. And so it was thought that Telenomus was never established in Florida. Well, surprisingly, in 2009, my lab were looking at a cattle farm. So this is all pasture grass, a type of Bermuda grass in this case, in the county just west of us, southwest of us. We were looking for something else, and surprisingly, we found Telenomus remus. And we're like, wait, we know they were released down here. How did they get here? Well, the thought is that, I told you that they were quarantined at a TPI facility here in Gainesville. There's unpublished reports that some of the workers in Gainesville released Telenomus here in North Florida. So that must be where these populations came from. I don't think they since they didn't establish here, I don't think they moved north. So I think those populations came out of Gainesville. Well, we're continuing to look, and let me go back real quick. So I have, using sentinel egg masses, I've looked all through North Central and Central Florida. Everywhere that I've put out egg masses, I found Telenomus. Last year, for the first time, I found them down here in Southwest Florida, but surprisingly, over several hundred egg masses, I've still never found them in Southeast Florida. I don't know what it is about that area that Telenomus doesn't like, but there's something there, and maybe it's competition from Chelonis. That's the only thing I can think of is there's strong competition, and that Chelonis has some kind of effect on Telenomus. But anyways, this is how we do our sentinel egg masses. These are two of the Borlaug fellows that were at Florida A&M University. This is Isabel from South Africa, Kennedy from the University of Zambia. They all came down along with Dr. Lekespe to help me put out the egg masses. These are from we have several colonies of fall armyworm. They lay on paper towels. We cut out the portions, pin them to the leaves, and then come back the next day or so, collect the eggs, and then either look for larvae that come out, meaning they were unparasitized, or about a week or so, 9 days later, you'll get Telenomus. So like I say, I can make a PDF of this. These are all the references that were in the talk, so people can go back and if they don't have some of these references, take a look at things. And I will now answer questions.
Okay, thank you so much, Rob. I think if it's okay, we can continue with. There will be 2 more presentations from the Indonesian side. So this is more of a discussion of 3 different presentations. So that's why the big title is actually about the journey from Americas to Indonesia archipelago. But it is very interesting to hear you mention that it has been detected prior 2014 from other countries, including Indonesia. That means that Spodoptera frugiperda has been in existence in Indonesia before 2019. And actually, if I'm not mistaken, the first time we detected or this report in Indonesia is like between 2018 or 19. So this will be a very interesting discussion. But I hope you can wait a little bit and we can continue listening to Pak Yayi's presentation. If Nazir, if you can put up the a little bit introduction of Pak Yayi. So our next speaker is Pak Yayi, and he received his PhD from Clemson University, and he's a insect molecular genetics, and he's been doing some research also on Spodoptera frugiperda. So it will be very interesting to connect what we found in Indonesia and what your research is in United States. perhaps in the future we can conduct a research together between Indonesia and United States. So Pak Yayi, the floor is yours. 20 minutes, please. Thank you.
Terima kasih Ibu Dami. Before I start with my presentation, actually I was going to point out the final slide on genetic diversity of frugiperda of Pak Rob that stated that it's been in existence in Eastern Hemisphere before 2014, which is very surprising. Where were we?
Yeah.
Right? We didn't know that it's already here.
Yeah.
Like we were 5 years late.
Yeah.
It's amazing.
That's
surprised me. Really surprised me. Anyhow, let's start with my presentation, which is actually I use a lot of Dr. Nagosi's paper, like most of us here in IPB, who's talking about frugiperda. We use a lot of the Dr. Nagosi's paper. So maybe it's not really new for Pak Rob and teams because we use your paper, basically. Okay, as we know, and also mentioned by Pak Rob earlier, that frugiperda is a native in tropical and subtropical region of the Americas. It's normally overwinters successfully in the United States, only in southern Florida and southern Texas. And the fall armyworm is a strong flyer and disperses long distances annually during the summer months. But it is recorded from virtually all states east of the Rocky Mountain. This is according to a report. But however, as a regular and serious pests, it ranges tend to be mostly in the southeastern. Actually, Spodoptera frugiperda is one of my subject when I was taking my Ph.D. in Clemson University a long time ago. But starting 2016, there is an explosion of Spodoptera exigua population in the Africa, especially West Africa, in Nigeria and Ghana. And the fall armyworm enters Sub-Saharan Africa carried by airplane traveling from the Americas. This invasive pest has begun to spread several countries in the world, such as India, Myanmar, China, Thailand, and of course, Indonesia. Availability of host and suitable environmental condition can be a factor in the spread of this pest. Considering these factors and geographical proximity to newly infested countries, this is certainly increases the risk of spreading frugiperda to Indonesia. These pests were first reported, I would like to stress, first reported in Indonesia in West Pasaman, which is in West Sumatra, in May 2019. And this is based on a report from the Ministry of Agriculture of Indonesia in 2019. And then, not from that, we made a trip to Banten, West Java, and we found, not West Java, Banten province. And we found a population of Spodoptera frugiperda then. It's in June 2019. But since then, frugiperda continues to expand to West Java, Central Java, Yogyakarta, East Java. Even in just a few months, it's already found in Eastern Indonesia, in the Morotai Island. But if after we saw the presentation of Pak Rob, so it is not surprised that it seems that frugiperda can fly very fast. I mean, in just a few months, it's already in Morotai, maybe because it's already been there since before 2014. So our study was conducted by conducting literature study on frugiperda, which is reported in agricultural land everywhere in the world. And the distribution factors and genetic diversity to be compiled in this paper and can be used as basic information for further action. And we use a lot of Dr. Nagasi's paper for this work. This pest was spread to several countries in the Americas, Africa, and Asia. This pest has been reported to attack several plantations of corn, sorghum, millet, and other grass groups. In 2016, there was an explosion of Spodoptera frugiperda in West Africa, and then it was reported that it entered Indonesia. Once again, our first report is 2019, then spread quickly to various regions in Indonesia. So Spodoptera frugiperda infested corn in Honduras and causes yield loss up to 40%. This is the report based on report in 2006. And then early 2016, frugiperda was reported in several West African countries, such as Nigeria, Togo, Benin, and Sao Tome Island. This report is the first indication of the establishment of population of this pest in Eastern Hemisphere. This polypagous nature of an ability to fly long distances of frugiperda pose a significant threat to African agriculture with the potential of rapid spread across the globe. This is according to Nagosi 2017. In Asia, the first reported frugiperda was in Karnataka, India, in May 2018. This is based on Desmukh et al. 2018. This pest infested more than half of the corn plantation in North Karnataka, with an average infestation of more than 50%. The spread of Spodoptera frugiperda was reported to extend to Yunnan, China in January 2019. And in Myanmar, report of frugiperda were found in several areas in Myanmar from August 2018 to January 2019. And on May 9th, 2019, the frugiperda was discovered in Nawalpur district in Nepal. Spodoptera frugiperda that was reported to have infested 15 provinces in China in 2019, and most of the crop that was damaged were corn and sugarcane. It is suspected that Spodoptera frugiperda that infested China came from Southeast Asia, such as Bangladesh, Myanmar, and Thailand. And as we know, our first report is in 2019 in West Pasaman. Then a few months later, we found it in Banten, western part of Java, and then in East Java, and then we found it in Morotai, eastern Indonesia. So what's the factor that play a role in dispersal of frugiperda? So in general, the distribution of Spodoptera frugiperda is largely influenced by the distribution of its host plants, such as corn. The climatic condition in the main corn producing areas are very suitable for the development of and reproduction of frugiperda. This shows that the report on Spodoptera frugiperda infestation in West Pasaman were due to the abundance of availability of corn in the area. West Pasaman is one of the largest corn contributor in West Sumatra, with corn production in 2018 reaching more than 360,000 tons. well on corn in new habitat, so it can be threatened food crops, especially corn. In Kenya, for example, the corn is cultivated all year round. Kenya is located in equator, so they have two growing season, and the main season is in March till July, while the minor season is in October till February. The availability of host throughout the season without planting breaks increases the risk of pest infestation. This is evidenced by the loss of corn production due to the pest infestation suspected as being frugiperda. And the differences in the growing season in the season are different from farmers in Ghana. According to Neboyin 2020, farmers in Ghana start planting corn in mid-July when rainfall is stable, and the trapping yield increased during the vegetative period until it peaked in August. The most common larvae are found in corn plant with 6 to 12 leaves. This is in accordance with the research conducted by Murua that Spodoptera frugiperda prefer corn plants during the vegetative stage. The abundance of frugiperda in corn plantation is influenced by the stage of plant growth, rainfall, and relative humidity. Temperature and humidity are the main environmental controls that affect the growth of and development of distribution of insect. Wang et al. Stated that the highest probability of the presence of frugiperda was at 28 Celsius. Frugiperda does not go into diapause and therefore cannot survive below freezing condition at any stage. The result of other studies state that the optimal temperature range of for the development of frugiperda were from egg to adult is between 26 to 32 Celsius. In South Sumatra, Spodoptera frugiperda can grow well on corn at a temperature of 29 and relative humidity of 74%. The parameter used to estimate the distribution of species is climate. One of the methods used to predict the potential distribution of species is the climax model. Climax combines the growth and stress indices of one of the species to see their potential distribution, which is described by the EI or Eco-Climatic Index value. The EI value described as a potential distribution of species in location. The EI value for the Indonesian regions ranges from above 75, indicating that it is very suitable for the development of frugiperda in Indonesia. And the direction of and flight distance of insect is greatly influenced by the wind, system. Wind pattern can affect the frequency, intensity, and displacement of Spodoptera frugiperda migratory flights. Migration flight direction is highly correlated with wind direction. And Spodoptera frugiperda has high flying power. The adult of frugiperda can migrate more than 500 kilometers. This is according to Prasanna et al. 2018. And when the wind pattern is appropriate, the imago can fly further. The adult frugiperda can fly up to 1,600 kilometers from Mississippi to southern Canada in just 30 hours. This is based on Rose et al. 1975. Another factor that has role in the dispersal of frugiperda is trade. Increased trade and and air transportation in Africa can be a factor in the spread of Spodoptera frugiperda to East Asia and Southeast Asia. Spodoptera frugiperda can be carried by aircraft, either in cargo containers or aircraft wheels. the introduction of frugiperda can be in the form of eggs, larvae, pupae, or adults, or even all stages of development. The distribution route of frugiperda can be spread without assistance and is carried along with commodities. Meagher et al. 2006 stated that insects in cardo were most often found in cut flowers, plant parts, fruits, and based on its feeding behavior, it is most likely that the introduction of frugiperda larvae from America was in plant parts such as corn cob husks. And this section has been discussed by Prof, but I'd like to go over what we found here in Indonesia. Spodoptera frugiperda specific strain polymorphism were divided into two groups. This illustrates the asymmetric distribution among plant hosts in the field. Frugiperda strains, C strain and R strain. And C strain are found in large grasses such as corn and sorghum, while R strain are found in grasses such as rice and Bermuda grass. The C strain was reported to infect corn, sorghum, cotton, while the R strain infested rice, sugarcane, and grasses. However, the host attacked by frugiperda does not necessarily show its specific strain. Research by Lotto et al. 2019 found that frugiperda which feed on corn is an R strain. This is in accordance with research conducted by Sartyami et al. And frugiperda found in corn plantation in Banten shows the R strains. Research conducted by Juarez et al. 2012 showed that the strain found in corn plant were R strain. This is the table of genetic diversity of frugiperda. Geographically, the distribution of frugiperda in the Eastern Hemisphere is possibly from a population originating from Florida and the Caribbean, which is the FL type. Research conducted by Nagosi et al. 2017 found in frugiperda found in Togo, South Africa, and Karnataka, India are genetically related to Spodoptera frugiperda from Florida. Nagosi et al. Also conducted research on samples found in Myanmar, and the results show that 22 samples of frugiperda larvae from Myanmar were confirmed to be in the same group as frugiperda from Florida. All larvae sample of frugiperda C strain from Myanmar has similar results to those observed in India, Southern Africa, Kenya, and Tanzania. CO1 gene has been widely used for molecular identification of frugiperda, and Nagosi et al. Use CO1, and we use also the same CO1 primers in our work here in Indonesia based on Dr. Nagosi's paper. So in this paper, Nagosi identifies specific strains of Spodoptera frugiperda in Washington, Georgia, Florida, and Texas. They identify samples of larvae originating from Brazil, and the results show that it has a kinship with samples found in Texas and Florida. And in Colombia, CO1 is used to determine the proportion of C strain and R strain of frugiperda that feed on corn, sorghum, cotton, and rice. And the most commonly found strain is the C strain. And frugiperda strain found in India, Myanmar, China, and most of Africa are incompatible with their host plants. This is corroborated by research conducted by Juarez that found 16 samples of larvae in corn plants were R strain. And this discrepancy occurs because the relationship between the mitochondrial koi one. I usually call it choi or CO1. Gene and strain identified its identity is disrupted by interstrain mating. Mitochondria are inherited maternally. Mating between female R strain and male C strain will produce hybrid female offspring of R strain. This could be a factor in that some of the frugiperda found in corn plant is an R strain. Nagosi et al. Also conducted a phylogenetic tree analysis of 62 larvae samples from Togo, Africa. The construction of this tree based on the nucleotide sequence of the CO1 gene using neighbor joining method shown in the sample from Togo. So that the sample from Togo is in the same group as frugiperda from America. And the result showed that 40 samples of larvae were C strain, while 22 samples of larvae were R strain. And phylogenetic tree analysis was also carried out on sample of frugiperda larvae from Myanmar. And sample of frugiperda larvae were taken from 8 provinces, namely Yayi, Mon, Kayin, Naipi, Tau, and so on. And this phylogenetic tree of 106 samples of frugiperda from Myanmar identified 2 haplotypes as C strain and 3 haplotypes were R strain. So, in conclusion, Spodoptera frugiperda strains, C strain and R strain, have spread to various countries in the world, and the spread of frugiperda is influenced by climatic factors such as temperature, humidity, and wind direction, as well as human factors through trade. It is possible that for frugiperda to spread through West Africa, India, Bangladesh, Thailand to Indonesia. Phylogenetic analysis using the CO1 gene can be used to see the genetic relationship of frugiperda. The specific strain found in Indonesia from Banten sample are strain, but from other region it was C strain. So I think that's it. That's all for my presentation. Thank you very much, Ibu Dami.
Okay, thank you, Pak Yayi. Pak Yayi, I believe that there are data from Indonesia from several researchers, either Bu Dewi and also Fajrin. Did you include that?
Not in this paper, because this study was done before Fajrin. But I noted, I mentioned that
okay.
Using different primer, we found that the strain of frugiperda in Bogor is C strain, I think, if we use the CO1B primer.
Yeah, and so the work you did with Fajrin was also using CO1 and also TPI, right?
TPI, yeah, CO1A, B, and TPI.
Oh, CO1A.
Yeah.
Okay. So, Rob, just a little bit background. Actually, Yayi and his colleague, they've been working on the Indonesian fall armyworm, and they did several research in West Java, and then also other people have worked also in Sumatra. So there's a whole array of data from Indonesia, which unfortunately it wasn't shared here, but we can share with you the publication. I think the publication is in English. So I think it would be really wonderful if we can discuss what we can do together with that. So anyway, thank you so much, Pak Yayi. Let's just
one more thing, Bu Dami. Right now we have 2 more works in Bali and Nusa Tenggara Timur.
Okay.
We're working on strain of frugiperda.
Okay, so that's wonderful. We have this whole data sets of frugiperda strain from Sumatra down to Java then. And then also, I don't know if Kalimantan also have, and then it's going to eastward to Bali and then also to Ntt. So if you look at the map of Indonesia, you can see that we're trying to collect data from the most western Indonesia to the east. So maybe this is something that is of interest in the future. So a little bit, the last speaker, Pak Nazir, if you can just put the CV of Ihsan. So Ihsan is a faculty member at the University Muhammadiyah. And this is a little bit information up here for all the other participants who may not know Ihsan yet. So he finishes both master and PhD from IPB. And then he was a Burlough Fellow. And there, yeah, you can see all the awards and grant, USDA Burlough. And then he received several research grants for his PhD. And after he finished that, and he has quite a number of publications for a young faculty members. So it's very honored to have Ihsan here. So Ihsan will discuss about the biological control in Indonesia.
Okay, thank you, Ibu Dami. So everyone, it's been nice here. So before we start, let's release yourself. If you want to stand up for a little bit or take a drink, something like that, just easy. So Nazir, can I share my presentation now?
Yes, please.
Okay, thank you. Okay, first of all, I would like to thanks to Ibu Dami to facilitate this webinar, and also to Rob from US, even in holiday day, and also to all colleagues from US, from Philippines and another country, and also all participants. So today I'm going to talk about new association of natural enemy of exotic pests, Spodoptera frugiperda in Indonesia as a preliminary data. So it's been explained very well from Rob and also Yayi how Spodoptera frugiperda spread all around the world in 2 years from US to Indonesia. And since then, many research has been done in Indonesia, including detection and monitoring, genetic variation, biological control, and also ecology of Spodoptera frugiperda. And here, as research entomologist, I'm doing research on detection and monitoring.
Excuse me, Ihsan, maybe you want to put this up into the slideshow so that we can see the full slide.
Okay, I'm sorry. Okay.
Yes, thank you.
So based on this various research topic, I'm doing research on detection and monitoring, and also seeking natural enemy of Spodoptera frugiperda in Indonesia. So, yeah. So, but looking back to the biological control concept, there is a two type of biological control. That is classical biological control and also conservation biological control. So this is what we asking in this presentation. So in classic biological control, it's explained that exotic pests controlled by exotic natural enemy from country of origin. But it's different with conservation of biological control that local natural enemy conserved for biological control purposes. This is when Spodoptera come to Indonesia as invasive species. So the question is there any natural enemy associated with this pest, or can be the local natural enemy associated with them in the form of new association? But in the premise of classical biological control, it's explained that local enemy cannot control the exotic pests, while the damage done are far too great. Why? Because the new association between pest and natural enemy will not result in suppression, because the adaptation takes too long. And then based on this hypothesis, that hypothesis by Hokanen and Pimentel, what does the data say? Then we did a quick survey in Yogyakarta, the city where I live now. We did a quick survey on the present early detection on the population of Spodoptera in the field. And we can see in Sleman district and Bantu district as the most common urban area in Jogja, and also agricultural activity here, we can see that population size of Spodoptera frugiperda is different in each region. This is just by a quick survey, one-time survey. Then please keep in mind that this data is showing the infestation level. Even though there's some data showing the high population, we do not see any serious attack rate from the field. Therefore, we continue our research to study the population dynamic in some area. We can see here the population dynamic showing the same trend, but from all the larvae we collected in one growing season, we only collected one parasitoid. And I'm not sure whether it's parasitoid or not, because this tahinid fly emerged from one parasitized larva from sick location in Bantul, Yogyakarta. This was done in 2021, and then we wider our research area, including all area of Yogyakarta and also West Java, to survey natural enemy associated with Spodoptera frugiperda. Interestingly, here is different from raw data. We can see that Telenomus can be found in all locations of Yogyakarta. This parasitoid is the most abundant parasitoid. And then also we have Stenobrachon, Brachymelassus, and also Brachymeria femorata. And in just last few months ago, we also collected Trichogrammatidae, Platygasteridae, and also Eupelmidae. And another parasitoid, another natural enemy, we also found a mite, Tetranychus, and also Spodoptera. And one interesting finding is Megacelia scalaris. We found a lot of number Megacelia scalaris emerging. We're not sure they are emerging or not from Spodoptera frugiperda, but a lot of number of this fly in the rearing container. So we keep this data until this publication come from India, that Megacelia scalaris is the new parasitoid of Spodoptera frugiperda. Then compared to the data from Bogor, West Java part, here we also find another parasitoid like Aphanteles, Charops, Euplectrus, Microplitis, and Telenomus and Trichogramma. And also we found a predator like assassin bug and also larva of ground beetle. And from this parasitoid, we found also is same trend data that in Bogor, Telenomus is the most abundant and the highest parasitism rate among the other parasitoid. Then we test in laboratory because Telenomus has been established in Indonesia, and we use it for controlling Spodoptera litura, the previous Spodoptera genus in Indonesia. And then here we can see in our published publication, we can see that parasitism rate of Telenomus in parasiting Spodoptera litura and Spodoptera frugiperda is not really different. And when we try to rearing them using larvae feeding on natural diet and also artificial diet, they're also showing higher parasitism rate. It means we can use Telenomus remus as a promising biological control agent to control Spodoptera frugiperda in Indonesia in the future in other countries. Because to my knowledge, we also have report from another country reporting Telenomus as promising candidate for controlling Spodoptera frugiperda. Then when we compare the performance of some parasitoids, still Telenomus showing the high parasitism rate, SM value with Trichogramma silotribe, but this is still unpublished data with our team. And what we see from this interaction, we can see Spodoptera frugiperda can be parasitized by Telenomus and some another parasitoid. It means that local parasitoid who already established Indonesia can control Spodoptera frugiperda. It's also been shown in another interaction between leaf miner and its parasitoid. It's long story in Indonesia. Maybe after this, Budami can add more information. And also another story, the example of exotic natural enemy attack by local parasitoid, as shown by the gold fly, Spodoptera connexa, attacked by local parasitoid. Here is here, Spodoptera used to control a weed, Chromolena odorata, but in different area, they attacked by local natural enemy. So what does it mean from this here? If I look back to the data that we found in Yogyakarta, when we start the research in 2020, we only find tahinid fly and then 3 parasitoid and then more parasitoid in 2022 and also more and more parasitoid in 20 and 23. Therefore, another question has been generated, like association have not been formed at the beginning, and over time the association are formed naturally, maybe because at the beginning natural enemy is still not ready, or not, or how, and the new association of pest and natural enemy may occur even with a long time. So based on this finding, what does it mean? Then we need to revisit how can an implement a new association hypothesis, especially when we work in tropical country like Indonesia, where we have a high biodiversity. So last but not least, I would like to thank to IPB University for providing me postdoc program, to my institution who support me to do this collaborative work, and to Rob USDA, and also to Dr. Hasib, Dr. Legaspi, who facilitate me or who helped me to know Rob and another colleague from USDA. Thank you so much. So this is my contact. If you have any question, you can send me an email, or we can continue in this discussion. And I'm very open for any question and possible collaboration. Thank you so much.
Okay, thank you, Ihsan. Thank you so much. So, ladies and gentlemen, all participants, we've heard all 3 presentations. So let us start the discussion, and I think it's open to anyone, including if Rob wants to ask Yayi, or Yayi wants to ask Rob, and Ihsan wants to ask Rob. So this is more. I would like to make it open also for the speakers if they would like to ask each other, because this is an opportunity for us to comment, to react to each other's presentation. So I see that there are already questions that has been written here, or if you would like to ask question, you can also raise hand directly if you would like to ask directly to the speakers. Okay, but before that, I would like to give an overall, how should I say this, an overall, not really conclusion, but the highlights, highlights of what we've heard tonight. So it's very interesting to hear Rob's presentation. Well, I'm a biological control person, so I'd like to comment more on the biological control. Well, first of all, as I mentioned earlier, that in Indonesia, we've also looked at the genetic variation, and it seems that so far, what we have found in Indonesia is actually also the C strain, if I'm not mistaken, rather than the R strain. So I think this coincides very nicely with your data. And then for the biological control in Florida, it's very interesting also that you found the Telenomus remus as the most dominant, as we found also in Indonesia. And I did not know that Telenomus remus was introduced to United States from the island, whatever, I forgot that was, Caribbean or something. Mm-hmm. So what you mentioned that you cannot find Telenomus remus down there in the south of Florida, but it's already established in other places. Judging from the data from what Ihsan found about Telenomus remus in Indonesia and what you've seen so far, so do you think that, well, first of all, is Telenomus remus actually a natural control of FAW in the Western Hemisphere? And is it, why, if it's yes, why is it not in Florida? I mean, does FAW moved from the south to the north? I mean, this is something that I'm not quite clear yet. The second point that I want to find out or discuss with you, it's very interesting to see that if the frugiperda is new in Indonesia, but then it can be attacked by Telenomus remus, which is, maybe it's indigenous in Indonesia, but it can attack the new exotic pests. So with respect to that, what do you think about this new association stuff? I know that this is an old paper by Hokkanen and Pimentel when they mentioned about new association that should be looked at, because we've been focusing more on exotic natural enemy from exotic area, but we're not looking at new association hard enough. So when we found this in Indonesia, I thought, maybe it's time for us to look at new association between invasive species and local biodiversity. Maybe we haven't really looked at that. So anyway, Rob, just this quick 2 comment first, if you would like to.
Well, I think that, that earlier paper was more in temperate areas. And I think what Ihsan was saying about looking at it in tropical habitats is actually really important, because, there's other Spodoptera that are in Indonesia and all through, the Eastern Hemisphere. So I think a lot of these natural enemies, like Telenomus, will go to other Spodoptera species with no problem. There are Telenomus species in the Western Hemisphere. There's debate as to whether it's actually Remus or another Caribbean species, but at least the DNA work that Rod did, it looked, and our postdoc, from a DNA analysis, it was Telenomus remus, and again, it was released a couple times in Florida. Why it's not in the Belgrade? So when I say South Florida, there's southwestern Florida, which is kind of towards if you look at Florida, there's a big lake down in the south. So anything kind of west of that is considered southwestern Florida. And then south central or southeastern Florida towards Miami and Homestead is kind of a different area. And the agriculture is a bit different there. And Dr. Legespi asked, and I really don't know why Remus, why I haven't found it there. The only thing I can think of, and it's something we're going to test in the laboratory, is perhaps there's too much competition from Chelonis or maybe other egg parasitoids. But to be honest, I haven't even found other egg parasitoids, so like trichogramma. So it's kind of a curious result. But Remus is, they've released it in South America quite a bit. There's actually some really good studies in South America where they actually show the length of, how wide they disperse, how many you need to release to get control. And so I think it's like a lot of natural enemies. It's not going to control it on its own, but it's just a part of as they say, a part of the toolbox. It impacts some mortality to the population.
Okay, thanks so much. That is very interesting. Okay, I see Jesusa also raised her hand. So if I would like to give time to Jesusa, and then later on, I think Dr. Hasib also, if you want to ask a question. But now, yes, Jesusa, please.
Thank you. Thank you, Rob, for your insights. I was just wondering, because living here in Florida, it's noticeable that in South Florida, as you mentioned, the Miami area, they have different types of agriculture. There's a lot of big commodity growers with large acreage of just one particular crop. And they spray a lot of chemicals as well in those areas. As you go north, as you move up north, central to North Florida, where I am, it becomes more diverse. There's less agriculture, less big commodity. growers and less spraying of chemicals, just because of the agricultural landscape in the area. So that might be a possibility of why there's more telenomus in those landscapes. So that could be a factor. Biodiversity, the agricultural complexity, the landscape. Those may be reasons for why the natural enemy complex is also different. But that's just a speculation. I think we need to do more research on those, and also tying it with DNA studies to correlate the natural enemies and the pest. And then just a quick comment about your statement on association of these natural enemies and exotic pests. Something I'm sure Dr. Meagher is familiar with this. We both worked on sugarcane. In the 80s, there was the Mexican rice borer that arrived from Mexico to Texas, to South Texas. And so many different natural enemies were collected from all over the world, and they were released in South Texas. And I monitored these areas where they made the releases many, many years later. And I found there were just a few species that were established. And what was interesting was the ones that established were actually the native parasitoids that were already in the region. So it's obvious that the native natural enemies can do a better job in controlling the exotic pest. So that's just something to think about when we're doing conservation biocontrol, or you're looking at classical biocontrol, is to look at the native natural enemies. It may take a little time for it to actually be a really good natural enemy, but only time will tell which one would do the best. But in the meantime, you could still do the classical biocontrol if that is something that would work better. So I'll stop there. Thank you.
Maybe can I add more opinion?
Sure. Thank you, Jesus. Thank you. Ihsan?
Yes, I think when parasitoid established come to new place, beside the landscape condition itself, the bioecology of its parasitoid itself affect the success in establishment of this parasitoid. Because when parasitoid come to the new place, there is a certain mechanism in regulating the sex ratio, as our previous paper with Dr. Buchori. I'm working in Apanteles. They very easily to switching the sex ratio. In some case, they're not producing the female. So that's why they do not establish in the next season. But recently when Rob find telenomus recently in another part, maybe it's related to the report, how the Spodoptera enter from some ASEAN country. Maybe some telenomus included with Spodoptera frugiperda in egg or something like that. So beside the landscape itself, I think the bioecology of parasitoid itself also affect the establishment. And in my opinion, I think in Miami the wind is very strong. I think it will swipe all the parasitoid in there. To me also, it's very strong. So for parasitoid, it will very, very. Thank you.
Rob, do you have any comment, or should we go?
I think we have a lot of questions here.
Yeah.
Okay.
There's many questions. If Pak Muhammad Hasib, if you want to ask questions yourself, or should I read? I think I would like to give the time to you.
I see. Thanks so much. I think all three, panelists has presented very well. When we deal with invasion and new pests moving from one ecologies to one country to island, is really opportunity for all of us to work together to find solution. Work with extension agent, growers, and other. Some of my thoughts were, I think I requested Rob, and Rob replied to my question that now this insect is moving in many places in the Pacific, for example, Indonesia, many other countries in Africa, and so forth and so on. And United States has solution against this, pest in case of either biocontrol agent we are using, or we are using hybrid varieties. Certain countries might not like to import hybrid varieties for, to be released for farmers, but they, use them as a commercial, for food and other feed. So one of the challenge we in the United States researchers are now feeling that this pest is in control. And if it's moving in many countries, many ecologies, and like in China, Indonesia, Malaysia, many countries use different practices to control it. So either by pesticide, biocontrol, and other practices. And then there are many different varieties. So we think a new strain might pop up for us. A lot of researchers are now working on it. Rob indicated that one of the species, one of the strain which we have from West Africa is a hybrid between CC and RC, like in between rice and corn and vice versa. So we feel that we might get a new strain, and then we need to work all together to find way to control it. It can be aggressive in case of plasticity, in case of reproduction, in case of climate, type of tolerance. So that part we need to also be looking into. The second thought is that, while this pest is in Indonesia, how your regulatory agencies are trying. For example, in here, we try in Florida using the strategies which Florida Department of Agriculture Consumer Service or FS, Animal and Plant Health Inspection Service have. And a new pest comes in, I think what they do, they try to use phytosanitary or containment or eradication procedures. So in Indonesia, I would like to learn from Ihsan and other that how when new pest comes in, how it can be contained and how you work with growers and other to depart training to these folks, whether there are certain procedure for that. If so, I would like to learn. Thank you.
Okay, thank you, Dr. Hasib. So there are questions to Ihsan, and also there's a comment to Rob. Maybe Ihsan, if you would like to answer, and then I think I would like also to give the time to Pak Yayi, if Pak Yayi would like to add on the Indonesian government. Ihsan, please.
Yes, I'm trying to answering the question I have in column. Thank you, Dr. Hasib, for the question. Here I also including your question. Yeah, some grower in Indonesia, they care about this pest, but based on my experience, some of them recognize this as Spodoptera, but when we inform them that this is the new pest, because the farmer in Indonesia, basically the farmer has a low educational background, so they do not know about the pest identity. So when we teach them how to recognize, how to distinguish between the Spodoptera and Spodoptera frugiperda, and they know this is the new pest. And so far, they spray feed to control the egg. They know how to control they know the best decision is to spray the egg because the egg will not hatch, so it's not harm to the plant. But some farmers also using like biopesticide, using fungi or plant growth promoting rhizobacteria in Jogja. They're using something like that too. And also here, some of my our alumni working in pesticide company, and we are working together to give some extension activity to help the farmer in facing this pest, how to control, how to conserve the natural enemy, and what another activity that we can do so we can control or manage this pest in more sustainable way, something like that.
Okay, thank you. But yeah, do you want to add on how do the government contain the spread?
Okay, thank you, Bu Dami. Yeah, in 2019, we were shocked that we have Spodoptera frugiperda here in Indonesia. Which is, well, the shock was 5 years late, I guess. But still, at that time, it was very hot topic. Reports were coming from all over Indonesia, and most of us or most of the farmers didn't know how to deal with the new pest. We didn't have any pesticide registered for that species. But I think the pesticide company worked faster than us. So, and now I think frugiperda is not a very hot topic anymore. It's there, and the farmer also realized that the damage mostly occurs during the vegetative stage, and they said the plant recover. I don't know how much of recovery after the damage, but it just maybe it's still hot topic for us, but I'm not sure in the field. Dr. Hasib also asked if we can contain this species. I think working together with pesticide company doing the field school, I think that's one way. My lab is still working on, not working on, but still searching for virus, because we have not, at least my lab have not found any NPV on frugiperda. We're still working on fungi. But I think that it's what happens is like we naturally heal ourselves. Well, I don't know, but maybe I don't know.
Yeah. Thank you. I'm not sure about this healing ourselves, but there are several. Maybe the natural enemy has.
Did somebody in power really work on it? We know that there's a IPM on at work.
Yeah.
Naturally.
So it's really interesting how in 2019 Indonesia was just shocked, and then you have all these pesticide company trying to come in with their new pesticide which has not been registered yet. So all of a sudden you have this flux of pesticide company trying to register their pesticide because of the fall armyworm. So it took time for that to happen, but that in the meantime, the Indonesian researchers also tried to find local natural enemy, which was not a lot at that time. So looking at what is happening now, I don't think it's contained. It's already spread everywhere. But it's interesting that the farmers seems that they're not really, I don't want to say they're not bothered, but it seems that they can overcome.
Like I said, it's like we heal ourselves.
Yeah. So anyway, this is interesting to be looked at again. So Rob, do you have anything to add before I'm going to give it to Diki, who's been very patient waiting?
Yeah, the only thing is in West Africa, they found the same thing. When it first got there, it was extremely important and everybody was kind of freaking out. But after about 3 years or so, they're like, yeah, it's just one of the things we have to put up with. They're not overly concerned like they were initially. So I think you're seeing the same thing in Indonesia.
Okay. Wow, that's good to know. Okay, Diki, you raise your hand. Diki Dwianto. Yes, please.
Okay, thank you very much, Prof. Dami, and everyone can hear my voice.
Yes.
Okay, so thank you very much, Dr. Robert, for your presentation. It's such a nice work about genetic differentiation of fall armyworm, Spodoptera, frugiperda. However, I wonder how to train from the same species the different host plane, like some individuals prefer corn or rice, that cause this phenomenon, or something that you mentioned before, that there are two populations, Florida and Texas population, that separated by the mountain range near the south coast. And
Yes.
One question for Dr. Yayi. You said that some places had been reported for this invasive species like Sumatra, Java, and Maluku, but you don't mention the island like Sulawesi. But so this is interesting, since if this artifact sampling can be filled and we will get a comprehensive data about the genetic differentiation of this fall armyworm in Indonesian
Okay.
Pelago. Yeah,
Thank you.
It will be wonderful. Thank you very much.
Okay, thank you, Diki. So the first question is to Rob. Please, Rob.
So the two strains, the two what we call sympatric host strains, the C strain and the R strain, that's a little bit different than the geographical haplotypes that I was talking about. Those are the Florida and the Texas types are only within the C strain. Dr. Nagoshi has not found those same markers in the R strain. So we assume the R strain is doing what the C strain is doing as far as migration, but we don't know that for sure. So it's two different genetic differentiations within the same species. One is related either to host strain habitat and or mating, and that's C versus R. And then within the C is the geographical haplotypes, the ones that affect migration, and that's Florida versus Texas.
Okay, thank you so much. Pak Yayi, Sulawesi, will we have?
Yeah, terima kasih. Thank you, Diki Duyanto, for the question. Well, actually, the thing that limited us from working for more data is just resources. I think as soon as we have enough resources, we would like to collect as many data as possible from as many provinces in Indonesia, because we know that from our previous work with Fajrin Budami, we also would like to see the correlation between vegetation around main crops in correlation with frequencies of strain. We would like to learn about that too. So now we are working on collecting data from as many provinces as possible from Indonesia. Just we're just waiting for resource. I think that's the main limiting factor for us. But soon we're gonna have that. Hopefully. Thank you. Finger crossed.
But it's there's this question from Pak Hasib, who's actually been answered by Rob. But I was wondering if Rob finds that there's a strained within strain hybrid C over C and also between C times R. Maybe this is something that we should also look at in Indonesia,
Yeah, I think so.
And there are hybrids also in between
Yeah.
strains.
Yeah, we have the know-how to do that. I think we can continue fighting work, and we can have more data on that too.
Okay.
Yeah.
And we can collaborate with Rob from USDA. Yeah.
I really hope to, because Pak Rob, I work at USDA Maryland for 4 weeks when I was taking PhD. Actually, I'm Dr. Shapiro's student, Dr. Martin Shapiro. Yeah, he's my professor when I was in Clemson. Yeah.
Actually, a lot of the strain, the within strain, the C by C hybrid, I just learned about. I was in Africa a couple of weeks ago, and there's a really good lab out of Montpellier, France, that's doing some interesting work, and they're the ones that are coming up with that there's perhaps some differentiation within the C strain, kind of a centralized Mexico group and then everybody else. So this is new research. It's not out of our lab, it's out of the lab, and I can give you that information if you'd like to see how they're doing that work.
So Rob, if there's a hybrid between strains C times R, do you think that will be more voracious strain because they can attack everything, or what's the prediction here?
No, not necessarily. What I think, the funny thing that's important with it is what marker you use to separate strains. If it's maternally inherited and everything is, all the females are C strain, but the males are R strain, you're gonna pick that up as a C strain. You're not gonna pick it up as an R strain. So that's why I think the TPI gene, even though it's more expensive to use that marker because you have to do sequences, it is more important and it's going to give you more information. And I think a lot of the early work in Africa by some other labs, they just looked at the mitochondrial, CO1 gene and didn't look at any type of genomic like the TPI gene, and that's going to skew your information. It's going to give you, that there's more C strain in the area than there probably actually is.
Okay, good to know. Okay, this is one question for Ihsan from Laila. I think you already answered it, but it's okay if I read it. What plants can become a place for natural enemies of frugiperda to live? And is it quite easy for farmers if implement, because farmers in Jember are starting to have frugiperda also, and even though it's still not at that economic threshold, they're still here. So what is the best place to find natural enemies? Okay, Ihsan.
Okay, thank you, Nurlaila Magvira. So I already answered here. In general, if we want to conserve natural enemy in the field, so we can start to planting flowering plant around the field. So it's the best plant to keep the natural enemy stay in the field, because in the absence of host itself, so natural enemy can still have a food source to support their lives, so they can establish and can attack the host when the host is available. I think so, Budami.
Okay, thank you, Ihsan. This is one good question from Wagner. Is this Pak Wagner from IPB? Well, anyway, let me read his question. Or do you want to just ask the question yourself, Pak Wagner? Okay, if you're not there, I'll just read it for you. Okay, is there a plan to start rearing natural enemies at a large scale? A very good question. And Ihsan answered that it's Dami's lab is going to do, but not a large scale. Well, to be short, of course, we would love to have it at a large scale, but no, not yet, because we haven't finished all the research on what is the best natural enemy that we can pursue. So it's still an ongoing research. But then, because of this question, I would like to ask Rob here. Does USDA already rear natural enemies and release it in the field for frugiperda?
No, they back in the 1980s, they reared a pupil parasitoid and did some releases, and it wasn't really that effective. They also reared a larval parasitoid from Bolivia. So, but again, it became established and then died out. So I guess officially then it's really not established. But no, I mean, that's the thing with frugiperda, especially in the Western Hemisphere. But I think you're finding it the same in Indonesia. There's a lot of natural enemies that will attack it, and so it's not that there's nobody around to hit all the different stages. I think what the important thing is the conservation biocontrol is to not spray over top of the natural enemies and to have some type of either flowering plant or another host where you're getting a feeding host that's not going to damage your crop, but will be a host for the natural enemy. Something like that there where they can then move over into your crop. And so I think maintaining the natural enemies you already have is going to be better for you, the research that you do, than trying to come up with something exciting and something new.
Okay, yeah, that's very good insight. I totally agree with conservation biological control. It's the way. But right now I don't know, because the farmers are still spraying a lot there in Indonesia. So that's the challenge that we need to face. Okay, I see Jesusa raised her hand again, or is this do you want to say something? Okay, yes, please.
Thank you. Yes, you were wondering about Dr. Tavares. His name is Wagner Tavares. He's from Brazil, but he works there for a private company.
Yes, for IPB, I think eucalyptus.
Yes. But now he's moved to another company somewhere near your where you're building your new capital. So he's not working there, but he has expertise in rearing parasites. So he could be a good contact, because that's what he does. He builds the biocontrol lab for the company so that they release natural enemies. And so I just wanted to give, he may have already stepped out. So he spent a couple of months in my lab too, in 2014.
Okay.
Yeah. So
thank you.
Just wanted to say that. Yeah.
Okay.
Thank you.
Thank you for the
information. Before I go, I'd like to just congratulate all the speakers. They're very good presentations, and the webinar is very informative. So thank you.
Thank you. Thank you, Jesusa. Nice to have you here. Okay, I think we've gone more than our 1.5 hours, and we're actually going towards the 1 hour 45 minutes. So I think we can end for tonight. As what Jesusa mentioned, I would like also to thank all of the speakers. Big applause for all the speakers. It's really wonderful to link what USDA has done and then what we in IPB are currently doing, and Ihsan also at UMY in Yogyakarta are also doing. So for species, exotic species, invasive species such as fall armyworm, I think it is very important for us to know what research has been done and how is it that we can collaborate and learn from each other, especially to overcome the attack of this invasive species. And listening to tonight, it seems that there are still more research to be done, and especially in the biological control area, as what we have seen from USDA, if you do not spray, it seems that this particular parasite, the Telenomus remus, if I'm not mistaken, is really promising. But then there's this Chelonos, which is, it's there, but I don't know, did we find Chelonos in Indonesia, Ihsan? No, okay. So that's good.
Only Telenomus, the same species, I think.
Ah, okay, yeah. And we have other larval parasitoid that we haven't actually looked at into more detail. But of course, that means it's a good opportunity to look for the future research. And then Pak Yayi will continue his work toward Eastern Indonesia and also hopefully for Sulawesi. And looking at the hybrid will be interesting to look at in the future. So, I see a raised hand by Pak Muhammad Hasib. Would you like to say something?
Yes, thanks so much. I really enjoyed the dialogue. Thank you for organizing. It was excellent. One of the things we do at Florida Agriculture and Mechanical University is, invite colleagues worldwide to come and join us. For example, Dr. Ihsan Nurkomar from Java Island at your university came here, and it was a pleasure. And we are still collaborating with him. We are writing different type of project under USDA Foreign Agriculture Service and other. And I then collaborate with Dr. Legaspi, Dr. Rab Mahar and other, and we jointly train, some of the folks coming from any part of the world. Mm-hmm. So here I invite any of your colleagues and student who can join us in Florida A&M University for shorter period of time, 3 months, 4 months, 5 months, to come and work with us in our ecology in Florida. And probably we also need to visit you. I recently visited Dr. Nurkomar, and it was excellent experience. It was first time going there, and I really like to see all different diversity of rice, cassava, coconuts, and so forth. And so I'm looking forward to collaborate further, and this is excellent, venture to support students and other. Thanks so much.
Thank you, Dr. Hasib. Actually, we have one faculty member who's looking for a PhD program. So if it's possible, looking into the future, maybe we can also send one of our junior faculty to your university and to learn. So that's one possibility that we can do.
Excellent. Thank you.
Yeah. Okay, so everyone, thank you so much. Rob, thank you very, very much for taking the time on your liburan. What is liburan? Holiday. Yeah, we appreciate and we really enjoy your presentation. It's very important for us to learn firsthand from the person who actually did the research. Thank you so much. And then also Pak Yayi, thank you so much for presenting the history and where Indonesia is right now. And of course, Ihsan, also thank you so much for discussing and making us think again about this new association and the role of natural enemy, local natural enemy that hasn't been really looked at in the past. So I don't know if we've taken picture. Hopefully we've taken picture. Pak Nazir, should we take picture together? Everybody opening up their, okay. Pak Rob, this is our certificate to you. This is what Indonesians do, Pak Rob. We always present the speaker with the certificate. So here is the certificate for Dr. Robert Marr. Thank you so much. And then the next certificate is for Dr. Yayi, I think. Thank you so much. Yes. And then the last certificate is for Dr. Ihsan Nurkomar. Yeah, thank you so much. So do we want to take pictures together, Nazir, or have we done that?
Yes, please, Udami.
Yes, okay. So if everyone can just wait for 1 more minute, we can take picture for everyone. And please, everyone, open up your camera so we can take. Again, Rob, this is Indonesian. We like to take pictures. So let's do this once and we can share with you. Okay, if you can count, Pak Nazir.
Yes, we have 6 screen for the first.
Okay.
1, 2, 3, and then the second screen. 1, 2, 3, And then the third, 1, 2, and 3. Then the fourth, 1, 2, 3. Then the fifth, 1, 2, 3. And the last screen, 1, 2, 3. Thank you, Bu Dami.
Thank you, Nazir. Thank you, everyone. Have a good day from people in the United States and everywhere. And good night for all in Asia. Thank you. Goodbye.
Goodbye.